volunteers who are parents of children affected by SHINE syndrome. Our mission is to provide the quality of life for children and families affected by SHINE syndrome through supporting collaborative research, such as today, among international medical professionals, nurturing a family community, and raising worldwide awareness. The researchers who have worked with us and agreed to give the presentation for SHINE syndrome in the Science of SHINE DLG4 webinar series have kindly volunteered their time to help us parents, caregivers, and patients understand the DLG4 gene variant and the tremendous amount of work and expertise required to go from recognizing a very rare disorder to understanding how it causes the disorder and identifying and developing therapeutics to effectively and safely treat the disorder. Today is the first of several webinars in the Science of SHINE DLG4-SHINE series. The format will be a 15 to 20-minute presentation by experts in the research who will present their current work in an understandable way for a patient caregiver audience. I have a few ground rules to get started first. One is that the researchers have very kindly volunteered for this, so please be kind back. The question and answer session should be kept in their scope of knowledge and may or may not be able to answer questions outside of that scope. If that's the case, we'll try to address it at some point in whatever way we can. So that includes insurance questions, costs, things like that, that they may not be able to answer for you. Number two, I have everyone come in on mute, and at the end of the presentation, if you could put your questions in the chat, and I will ask them of Dr. Tümer. Several questions have already come to me via email that I'll ask first. At the very end, SHINE Syndrome Foundation representative will speak about opportunities for involvement if you'd like that. With that, let's get started. I'm going to introduce the presenters and then let Dr. Tümer get started. Today, we are honored to welcome Dr. Zeynep Tümer and Amanda Levy. Dr. Tümer is a leading figure in the field of clinical genetics. Dr. Tümer holds an MD and a PhD and works at University of Copenhagen. She is a professor in the Department of Clinical Genetics at the Kennedy Center at the University of Copenhagen. Among her many achievements, Dr. Tümer was a main author on the groundbreaking publication that provided crucial evidence linking DLG4 gene variants to neurodevelopmental disorders. She has over 240 peer-reviewed publications, and her work has significantly advanced our understanding of genetic disorders, particularly DLG4 synaptopathy or SHINE syndrome. Please join me in welcoming Dr. Tümer as she shares her insights and research with us today. I'd also like to welcome Amanda Levy. She is a rising star in the field of genetic medicine. She's currently pursuing her PhD in cellular and genetic medicine at the University of Copenhagen. Amanda is already making significant contributions to our understanding of neurodevelopmental disorders. She has co-authored several important publications, including research on the DLG4 gene and its role in synaptopathies. She has a strong background in molecular biology and genetics. Her work is paving the way for breakthroughs in the future in the field. Please join me in welcoming both Amanda Levy and Dr. Zeynep Tümer. So I'm going to pass it on to Dr. Tümer.
Thank you very much for your kind words and introduction. It was so nice. And as Sabrina said, my name is Zeynep, and I will just start telling about, just a second, if my, I just have to figure out what is going on. Yes. I will start with an agenda so that you know what I'm going to talk about. And first, I want to tell about what we have been doing regarding DLG4 until now, the published work. So, and the other thing is that I want to tell a bit about synapses and how DNA-RNA protein relationship is. And I want to talk about DLG4 variants and how they affect what is the predicted effect on the protein. our research activities very shortly and the databases we are doing regarding that, and then there will be time for questions. So this is where we are. Denmark is a very little country, and we are in the cities, the main city, Copenhagen. It's a department of 150 employees, and my DLG4 research group is very little. It is me and Amanda at the moment, but we are trying to achieve as much as possible. So my relationship with DLG4 started indeed in 2017, where we have identified a DLG4 individual in our department. And then from 2017 to 2021, it took us, as you can see, 4 years to collect genetic and phenotypic information of 53 patients, and this was published. And DLG4 gene, as you know, codes for the PSD95 protein, which I will also go into the details. And after this 2021 publication, we had some, in 22 and 23, we have published a review to understand generally PSD95 and its interaction partners, how they are involved in neurodevelopmental disorders. We made an online gene reviews mainly for the clinicians for how they should kind of take care of the SHINE syndrome children and adults. We made a single gene publication, which was a very interesting variant. And then lastly, at the end of last year in December, we published a paper describing the epilepsy phenotype in DLG4. So with that introduction of what we have done, very shortly about the neurons and synapses. So in our brain, we have about 86 billion neurons, and the neurons are connected together with synapses, and we have about 1 quadrillion synapses in our brain. And PSD95 protein is in one of these synapses in the glutamatergic neurons. These are the neurons which excitative neurons, which give a charge to the nerve cells. And if you look at synapse in more detail, this is where PSD95 is, and there are a lot of proteins, which is called, this is the synapse, this is the presynapse where the charge is coming, and this is the postsynaptic region. There are a lot of proteins working in this postsynaptic region. Probably there are 400 to 500 proteins there. And PSD95 is one of the most important proteins, kind of both putting things together as both functional and structural roles in the domain. And it binds to a lot of receptors, which are very important and known for giving, for example, epilepsy on the membrane. And inside the body of the neurons, it also binds to a lot of other proteins, which are also, you may have heard the names of, for example, SYNAP protein, SHANK protein, SAPAP, etc. And if you look at the domains of or the functional parts of the protein, there are 3 protein domains which binds to several receptors and also other structures in the cell, and 2 other domains, SH3 and GK domain. Especially the PDZ domains are very involved in AMPA and NMDA receptors, which are also involved in epilepsy disorders. And how is so this relationship with the gene and the protein? So this is a general kind of cartoon where we have in the nucleus of the cells, we have the DNA, which are structures as chromosomes. And on the chromosomes, we have the genes. And genes have exons and introns. And exons are the coding part of the genes, and introns are the parts which binds the exons together. So these are between the exons. And what happens is it is the DNA, and the DNA from the DNA exons kind of splice together, and they make the part called RNA. This is called transcription, and the RNA goes out from the cell nucleus into the cell plasma or cytoplasma, and here the mRNA or RNA translates the knowledge of the exons to the protein. So this is the general mechanism. So our DLG4 is here, and the protein PST95 is here, so it is in the postsynaptic regions. And everybody has 2 copies of DLG4, one on each chromosome 17. So it means that we have 2 DLG4, which codes for half of the PST95, one of them, half of the PST95, the other one. And in DLG4 individuals, only one of these copies are mutated. We have a variant, only one of them, which affects the function of the PST95. And the other one is normal. So we have the SHINE syndrome individuals have one normal copy of the gene and the protein. So what happens is that when something happens to the DLG4, there are different types of variants. The change is called mutation, and mutation ends in a variant. And these are different types of variants which have different types of effects on the protein, which I'm also going to tell about. What we know for DLG4 there are 2 main types of variants, very roughly. One of them is the missense variants. I will also tell a little bit about it. And the others are called, in general, protein truncating variants. This is a general knowledge because I have also made we have made some cartoons to explain what we mean with that. And these can be divided into 2 main groups, nonsense variants and frameshift variants. Frameshift variants means that the reading frame, the protein is not read correctly, and the frame shifts and it ends in a mutation. It's in a variant. What happens is that these are the changes happening on the gene, and what happens to the gene has an effect on the protein. And it is generally, if you don't make research, this change is only a predicted change. Missense variants, if you go to the protein truncating variants, they normally end as loss of function mutations or haploinsufficiency. So if you look at this protein, this is the normal protein. If it loses its function, it cannot be used anymore. And there are 2 other types of protein deficiencies, which is called gain of function or dominant negative effect. Gain of function is the protein gains a new function, and dominant negative effect is also new function, but this time the protein has a bad effect on the normal protein also. And missense variants can indeed be any one of these ones. And in general, it is difficult to predict the protein effect of the missense variants, which really necessitates functional studies. So this is what we made together with Amanda. How can we explain the variants? Because genetics is indeed very complicated in a metaphoric way. So the alphabet of the genes or the DNA is ACGT. So we have only 4 letters to explain everything. So we made a sentence, which is the normal sentence, and it corresponds to the PST95 protein. So the sentence has a start, and has an end. And it gives more or less a meaning. The fox ate the rat and the man dug till the end. So this gives a meaning in itself. The missense variants are a single change in one of these are called nucleotides, one of the letters. So let us say if the R changes to an H, it becomes a hat. So it kind of changes the meaning. It becomes the mismeaning. So these are missense variants. when we talk about nonsense variants, it is a kind of, you can remember, it doesn't give any sense, so it is nonsense. So what happens is that, again, a nucleotide changes, but this puts an E instead of, let's say, A, so the end of the protein comes before its normal position. So this is a truncated variant. So this truncates the protein. I will also show a picture of that. In general, frameshift variants are when you have a single letter deleted, missing, or some new letters added. What happens is that when this changes, let's say this T is out, then suddenly the meaning of the sentence changes. It shifts because when you take T out, then A is followed by E and then T. So the dog borrowed, and it comes to an end. So this is also a truncating variant because it shift the meaning of the protein, of the reading of the protein. This can also be any duplication or other kinds of variants. Some of the variants which are at the boundaries of the, I can show it here, at the boundaries of the exons and the introns, they can also affect as frameshift variants. And there are a lot of the DLG4 individuals also have frameshift variants because of splice variants. These are variants in the introns, but affect how the RNA is formed. So if you go to the protein part, so this is the normal sentence or the DNA, and this gives this normal protein. We don't know how the missense variants affect the protein, so this is a question mark. And these frameshift variants or the protein truncating variants kind of truncate the protein earlier than it should be. So they are truncated, and in general, these truncated cut proteins are degraded in the cell because the cell doesn't want to have them, doesn't need this waste material, so they are degraded in the cell. So that's the reason why it is X. So these are haploinsufficiency or loss of function, are predictions. Most of them are all these kind of proteins. So in DLG4 or in SHINE syndrome, most of the variants are these protein truncating variants. And there are some missense variants also I will talk about. So what I did was, I know that Sabrina told me that sometimes it is difficult to read the gene reports. So how is it possible to read the gene reports? With DLG4, this is gets a bit complicated because different laboratories use different versions of the gene. So DLG4 has several versions, but two main versions are called beta version and alpha version. And these have two different numbers. So in your genetic reports, you will have this number or this number. If you see this number, it means it is this form. And if you see this number, it is the alpha form. So what is the difference? Beta and alpha kind of code for two different versions of proteins. But the interesting thing is that alpha version is mainly expressed in the brain, so it is functioning in the brain, and beta is everywhere, less in the brain, but it is more or less everywhere. Alpha is also everywhere, but the dominant form in the brain is the alpha. And the other difference is that if you look at it, the last 19 exons are exactly the same, exactly placed at the same place. The beta has 3 other exons in the beginning, and alpha has 1 exon, which is somewhere here on the gene. And if you look how they code for the protein domains, you can see that these 3 exons, not exactly, but part of them, codes for this PDZ1 domain. These exons code for this PDZ2, 9 and 10 PDZ3, 12, 13 SH3, 1620 GK. The only difference is that numbering is different for the beta and alpha, but exactly the same sequence and coding for exactly the same functional domains. So beta is kind of different in the start of the protein. So I hope that it kind of helps a bit understanding when you read the genetic reports. Now I will show a bit complicated table I've prepared. So this is kind of a table you can use to read the variant from the gene report. And what I'm going to show is that this is the alpha isoform, the shorter one, brain specific. This is the beta isoform, the different numberings. And each table has the name of the exons. This is kind of, Sabrina sent me something related to the SYNGAP homepage. So I wanted to just kind of do it for our gene, but our gene is a bit more complicated. This is the coding part of the exons, and in your gene reports, you will always see a C dot. And this tells how this coding C is translated to the protein, so it always has a P dot in the beginning. So this is the same for alpha or beta, and I also tried to see which color corresponds to which domain. And I made two examples, so it will kind of help to understand how this table can be used. If you want to just see, you can use this table. And please give feedback if you want me to kind of improve it, if I can make it better, easily understandable. So in your report, you will see something like C dot 659 G to T, comma, or colon or something, P dot glycine 220 valine. And they will always write which number is used to tell this. So what you can do is that this is C59 and this is this number. So you have to go to this table. So 659 is somewhere here. And it codes for 229, 220. It is here. And you can also see 220 is here on this side, beta side. So this variant is in the PDZ2 domain. And it is also in exon 9. If you look at this one, C1231, C to T, PR 411 term, it means that an arginine, this is a nonsense variant, this is a missense variant. So it only changes an amino acid, but this stops is a termination or end of the protein, as I have shown on the metaphoric explanation. And if you look at it, 1231, and it's again the same one, 1231, it is somewhere here. And the amino acid 411 is between this one. And you can also see that it should be somewhere in PDZ3 domain. So it is in exon 12 and in PDZ3 domain. So the difference is that in this one, this variant has not truncated the protein, but the protein is there, but misfunctioning. So it's the missense variant, which gives a misfunction. But in this one, the protein is truncated here. So the rest is not translated. We have only this part remaining, and it will probably be degraded, almost for sure. So this is how we can read the table and how we can try to figure it out. But I'll also say it in the end. You are always welcome to write to me if you want to understand a variant of your child, how it is. I have also made for some parents some cartoons which will help them to understand the variant. So we can also have online meetings. Just please feel free to contact me or Amanda. So this is about the basics of the genetics, how DLG4 and PST95 relationship, and how we can kind of translate what is written on the gene report to the protein to understand. And this is we will talk very shortly about that because of the limited time, but these are 3 different or 4 different types of research we are making or activities we are making. So one of them is mainly to make the molecular diagnosis to understand what's going on, or a missense variant is pathogen or benign. And the other is clinical studies or functional studies, which will in the end can be used in treatment of the individuals. We are not directly working on treatment. So we have some studies with the RNA, remember DNA, RNA, protein, to understand how RNA is affected. We also make a new thing which is called DNA methylation signature. We have not started yet, so I'm not going to tell anything. But our main clinical studies are this is what we started with. We wanted to focus on epilepsy phenotype, sleep disorder phenotype, migraine phenotype. And also, this is also a need from the families, how the children are going to be when they are adults. So adult phenotype, and we're also working a lot on deep phenotyping. So getting as much as information about the symptoms and clinical features so that we can make both a genotype-phenotype relationship, but also this can be used in the natural history or clinical trial readiness when treatment is there. So this is something, a kind of infrastructure we are building so that we can help. And the other are functional studies, and I will let Amanda tell about the functional studies because she is the person who is doing that. And she'll also talk a little bit about what we are doing with the adult phenotype and deep phenotyping, because she has prepared 2 big databases which are called RedGap databases. So I will give the word to Amanda now. Maybe Amanda is muted.
Oh, okay. Hold on. Let me find her. Oh, here she is. Okay. You should be able to unmute
All
now.
right. Yeah. Can you hear me?
Yes.
Yes. Okay, great. Thank you very much. Yeah, so thank you, Zeynep. And yes, I would just really briefly like to talk to you about the functional studies we're doing and really what the aim of the different approaches that we have chosen. So as you can see, we are using 2 main approaches that we have written as primary mouse neurons and prism screening. And these are really to try to capture different ways of figuring out what the effect of the different variants are. So for all of them, we want to use the neurons and also just mouse brains to model the different variants. So we will choose specific missense variants distributed throughout the gene. And then we will also use one representative loss of function variant, because that is the majority of the variants that we see, but they should all have the same effect. And then we will use this imaging, which will tell us something about the placement of PSD95 in the cell to see if there's an effect there, because that should affect the function. We will also look at the electrophysiology, which means the communication between the nerve cells, because that should tell us a lot about whether it's functioning or not, and will also be easily related to what is known about epilepsy. And then finally, we will, for the prism screening, where we will look specifically at mouse brains, we will look at the brain development to see where the nerve cells that have some mutant PSD95, we will see where in the brain they localize, because that will also tell us a lot about the function and development in individuals with different variants in DLG4. So that's the main and very briefly explained approaches for the functional studies. And Zeynep, I imagine, yes, thank you very much. So as Zeynep also said, we have these two out of all the clinical studies we're doing these two large data collections where we're using this database infrastructure called REDCap, which is a secure database made for researchers to collect clinical data. And as you can see from these two screenshots of some handouts we've made, they look quite similar. So I would just like to take a few minutes to explain the difference, because the SHINE Foundation has been so kind to offer to distribute knowledge about these studies so that we can collect as many and include as many of you as possible in these studies. So the first one over here to the left, which we have called deep phenotyping, it's a study where it's similar to the study from 2021, where DLG4 related synaptopathy is really defined, and where the clinical and genetic features of 53 individuals were described. Here we just want to expand the number of included individuals. So we really want to include as many as possible, regardless. The data here should be collected from healthcare professionals. So some of you have probably already been talking to your clinician or a geneticist about this study because they wanted to collect consent. If you do not know for sure if you're already enrolled or if your child is enrolled, then please let us know, and we also hope to distribute this handout through the foundation, because then you can give this handout to your clinician, your geneticist, your neurologist, just whoever you think knows your child's history the best, and then they can contact us for participation in the study. And we really hope to include as many as possible. Right now, we know of 200 individuals, although we do not have complete data from everybody. But that will definitely really should give a much better understanding of the disorder, which, as you know, is it differs quite a lot between individuals. So the other one, which we have called adult phenotyping, over here to the right, it is a little bit different. It's more of a sub-project or subgroup, because here we want to focus specifically on individuals with DLG4 variants who are 16 years or older, and thereby more or less falls in the adult category, because that will give us a lot of information about the outlook of the disorder and how people, what the everyday life will be like, and also the phenotype there. So for this, we want to include the data directly from families, or for those well-functioning enough individuals with DLG4. And here we also have this handout that have already been distributed on different platforms and is still available, I believe. And please, if you or your adult child is 16 years older and you want to participate, then please go ahead and do so. It can be done through this link or through the QR code. And while there is quite a decent amount of questions, but that's really to try to capture as much as possible. And if you want to participate and you go to click the survey link, then please make sure that you include all the genetic and clinical information. I can see that there's a lot of clicks where people have just gone to see what is in the survey, which is completely okay. But sometimes they have been partly completed. The survey has been partly completed, but it's difficult for us to tell if it's done or not. And in most cases, it's for all cases, there's an option to leave an email if you want to be contacted, but that is completely optional. And for most cases, an email has not been left behind, which means that we cannot contact to find out if it's done, or we cannot collect any more data. And if there's a lot missing, then we will not be able to use it. So if you decide to participate, then just make sure that you include all the data that you want, and then click the submit button when you're done, just so we know that everything is as it should be. Because that will be really valuable to have an idea of what the outcome will be of DLG4 related synaptopathy. So yeah, just to sum up, it's 2 separate studies. Everybody who is in the adult phenotyping study can also participate in the other. It's just for the adult phenotypic study, there's a bit sub-focus on the everyday life, and if there has been a difference in the phenotype from when they're a kid to when they're an adult. And there's a difference on who should complete the surveys. So I hope it's also all explained in these handouts, but I hope that also just gives a simple explanation on the difference and makes it a bit clearer that it's 2 separate studies. Yeah. And I think Zeynep is now muted. And
Okay, you should be able to unmute now.
I'm unmuted. Okay. So one thing I wanted to add to Amanda is that the adult phenotype is indeed, we have carried out together with some of the parents and families, and it has been very thoroughly looked through. And also the other infrastructure RedCap database for the deep phenotyping is also, a lot of parents have also been through that. So we tried to kind of get everything you would need in these information, so that we hope we can use it. And adult phenotype is also will tell us about the disease progression from childhood to adulthood. So in that way, I think we did it a bit differently than the other people do, because it's not one time point, but we wanted to just see the progression of it goes as far as we can do it in a database. So very shortly, so all the research, our main focus is that it should benefit all the affected individuals, and all the research we and the others are doing are all complementary, and it will help us to understand the disease and its progression. As Amanda said, most of the variants we have are protein truncating variants. We have knowledge about 200, and probably 10% are missense variants. So I think there are around plus minus 20 missense variants, and the rest is truncating variants. And these will behave in the same way, but otherwise we want to, in our capacity, as we are a very little group, we want to include all the missense variants as possible in due time, but we started with some representative ones. And the last thing, which is for me, it is very important, our research wouldn't be possible if I didn't collaborate with the SHINE Foundation, because we really had and have a very close collaboration. And your needs are important for us, because we kind of, our research is driven depending on your needs. And I think it's very important for success. So for that, I also want to thank SHINE Foundation for supporting the research and what they are doing. And with that, I will thank you for your attention. It took a bit more time than expected. And as I say, genetics can be complicated. Please do contact us if you have questions you want to ask privately or want to talk with us. Thank you.
Thank you, Dr. Tümer and Amanda. We really appreciate your time on this. I'm going to go ahead and pose the questions that were asked, first from the emails, then from the chat. But I did want to say, Dr. Tümer has kindly offered to meet with individual families to answer any more questions. So you can email her directly and she can kind of let you know a little bit more about that. And if your clinicians, like your neurologist or your geneticist, want to become involved and are very involved in rare disease, they can also connect with Dr. Tümer either through you or by themselves. So, so first question, why is there such a wide variation in the phenotypes of kids that have nonsense DLG4 mutations that are theoretically haploinsufficient, thus producing theoretically the same amount of PSD95 protein?
Yeah, this is a good question, and this is one of the kind of the mysteries of the genetics. So we really don't know. I don't know where we can be clever if we can collect all information, all the 200. But the most plausible explanation is that the proteins do not function alone. So we know that DLG4 is mutated. There's something wrong there, but there can be other proteins and variants in other proteins that kind of compensate the function or makes the function worse. So our genetic background, apart from DLG4, has a great importance. But these are some factors, they are not directly, you can say, oh, this can be something important, because each individual has about 5 million different changes in all the genes we have. So it is very difficult to find out how this, but there are also some research for some other genes where you look at the gene, the variant, but you also try to compare the other variants the individuals have in other genes. But this can only be possible if we have thousands of DLG4 individuals. So I would say very shortly, what we have in the genetic background has an effect how the mutation affects the phenotype.
Okay, thank you. And then the next question is kind of piggybacking onto that. Why is there a wide variation in the phenotypes of kids with identical nonsense DLG4 gene variants?
Yeah, it is exactly the same answer. Probably something is compensating or making it worse, but we don't know anything about that.
Thank you. And the next question has to do a little bit more with gene therapies. Have any other rare conditions linked to scaffolding proteins had luck with any gene therapy that you know of?
As far as I know, there's not yet any FDA-approved clinical therapies, gene therapies, but there are a lot of research going on different levels, especially for the neurodevelopmental disorders. There are a lot of researchers who also try to find out how these different therapies can be put into come to the brain, goes through the brain-blood barrier. But as far as I know, there's no FDA-approved gene therapy for similar proteins.
Yeah, I just want to add that there are gene therapies for other genetic conditions. It's particularly scaffolding proteins, I think is what you're referring to.
Yeah. But there are some gene therapies. Yeah, that's true. It's the FDA approved, but especially for the scaffolding protein, working in the brain. It is difficult. There are some gene therapies for the retinal disorders, for eye disorders, because it's also easier to come into the eye and make the treatment. And you have to really know the protein and its function very well before doing many things.
Okay. There are some cases where 2 or 3 kids have the same mutation location. Is there any advantages to finding those for parents finding those other parents with similar cases, whether it be based on location or is type more important, like nonsense, missense, etc.?
We have, I mean, as far as we know, even the same variant, same missense variant, can give different phenotypes. So in that way, it is, I think, SHINE group as a group, defining the needs and supporting each other is the best way to go, because I don't think that you can compare the clinical phenotype of 2 individuals with the same variant. As again comes back to the other, what we have in our genetic background or in our genetic baggage.
Thank you. Sorry, there are quite a few questions, so I'm going to try to get through as many.
Sure.
I think you actually answered this question, but maybe a quick recap. What is a loss of function mutation and what is a gain of function mutation? And maybe a question, if a gene therapy is developed, will the same treatment be able to help all individuals affected by both?
No, gain of function and loss of function, as you can see that. I mean, this cartoon, you can see my screen, can't you? Yeah.
Yes.
This cartoon is very nice because this is a normal protein. When it loses the function, so it doesn't have any function. So it cannot use as a hook. And this one has an extra function. So in a way, for the epilepsy disorders, especially the epilepsy disorders are the disorders which can be treated because there are 9 different types of epileptic drugs. But some of them can be used for the gain of function, some of them for the loss of function. So in my opinion, different types of drugs and different types of approaches would be necessary for treating the gain and the loss of function. Because if you have a gain of function, you want to stop this function, this extra function, as this dominant thing, this toxic thing. When this is loss of function, then you need to give more protein, normal protein, so that you have enough function.
Thank you. Okay, moving on. Are there any cases where the individual has DLG4 mutations on both chromosomes?
No, not to our knowledge. Probably it won't be. I mean, I don't think that it is culpable with life. Probably. I know in mouse you can have the knockout. They call it, so you knock both DLG4 out, but it is always difficult to compare mouse with human when it comes to reality. But I have never explained, and I would say I would think that it won't be possible with compatible with life.
Yeah, no, that's what I've been experiencing as well, or other researchers have said too.
Yeah.
For SHINE kids, have we been able to determine if there are ancestral familial mutations, or are they primarily de novo?
Most of them are de novo, but we have some parents carrying the variants, and in some cases, I don't know how many, but we have a few. And in one of these, I can't go into the detail, but the parent had very few symptoms in childhood, and the young child has a bit more symptoms, but more or less fine. It was also a protein truncating variant, so it is also a mystery. But there are some parents, but very few of them. I don't know if Amanda, do you know? Do you have a number? I remember 1 or 2, but not more.
I don't know the exact number, but especially for some of the missense variants that we're more unsure of, there are more that are not de novo.
Yeah.
But it's not. I don't have the exact number. No.
And then if the missense variants which are inherited from the parents, we are not always sure whether they are disease causing or not. This is always, I mean, it is always a problem in the clinical genetics for the missense variants which are inherited from parents without any symptoms. So that's also something you have to carry out some missense functional studies to understand some of the missense variants.
Okay, so there's 2 more, so I'll finish off here. In addition to the REDCap data collections, would it be useful for you or the researchers to be able to have full access to the DLG4 case medical and mental health records and be able to directly interview observed cases if parents are willing to release from HIPAA restrictions?
Yeah, it can be. It can be useful. The only thing I would say that at the moment I do not have the capacity to do that, but I think it will also be something to be done when we have this thing. It can be also good to have some of these studies so that this clinical trial readiness, I mean, this is all we are trying to push for. So it is a good idea, and I think we should also think of that.
I agree. And so this is the last question, and it's more of a clinical question, so we can see if you can answer this or not, or have any ideas. Is there yet a sense how DLG4 variants might impact bladder function? We have had a very long journey with urinary incontinence with our daughter to date. Initially, the assumption was her difficulties in this area related to her learning ability. It has become clearer over time that this is not the case, but rather due to the physiologic function of her bladder. As a drug that relaxes the bladder has resulted in significant improvements to her incontinence. That's not something I've experienced yet from any parents, but do you have any idea or any sense of how it affects function?
I don't have that either, but I have also been looking at, because this protein is so important, and I couldn't find good publications trying to understand, because muscles and nerves make also connections. So it's not only 2 nerves, but a muscle and a nerve makes also a connection. And I have an idea that PSD95 can also be functioning there and may have an effect. But if you look at the results of the clinical spectrum, I cannot remember, but that is also, for example, this is something, it will also give an idea how the other children are affected with regarding the bladder function. But I cannot just remember from the top of my head about that.
Yeah, perhaps this is something that could be thought about with the mice that are being developed for DLG4 models.
Yeah.
And I think the parent that asked that, if they want to ask it in the Facebook group so we can get a sense of how many other parents are experiencing this as well, that would be really helpful. We have one more comment related to the bladder dysfunction. And they say this could be due to neuronal dysfunction in the bladder, affecting the contractility tone of the muscles in the bladder sphincters. And I think that's kind of what Dr. Tümer said as well.
Yeah, because the connections, the muscle, because muscles also needs nerving.
Right.
So it is, but for example, it is very good to point out to some important problems in the community, because this is why, for example, we started thinking of migraine, because these headaches, is it something common, does it have anything to do with epilepsy? Do they trigger anything? So, and they can be treated. So this is, for example, why we want to focus also on migraine and sleep disorders or related things. So it will be nice to know these common problems.
I agree. And I think, oh, somebody just put in another question. Okay, I have heard DLG4 is also expressed in muscle tissue.
Yeah.
Could this be tied to bladder control? So that sounds similar to
But
what you said.
DLG4 is, okay, the thing is that when we started, it was thought that DLG4 was not expressed in the blood. If you look at the database, which is called GTEx, it's a big expression database. It was not expressed or very little, but when we look at the blood, we can see that it's expressed. So we know that we have some normal individuals, some muscle biopsies, and we have also checked out. It is expressed very well in the muscle, expressed in the skin cells, fibroblasts. It can also be used, blood can also be used, even though it is not expressed very highly. Skin is better. And of course, it's also expressed a lot in the brain. And we also try to find out how these two isoforms are expressed in different tissues, but I didn't go into the details of that.
Well, I just want to say, I think it's kind of getting to be the end of the hour. So I know a lot of people have to go back to their families. And thank you so much for being with us and explaining all of this to us today. And I thought it was a really wonderful, wonderful explanation of it all to both of you, Dr. Tümer and Amanda.
Thank you so
much. Thank you. So I'm going to let Mara, if she wants to talk about any opportunities that may be available with the Shine Center Foundation to get more involved. And then we'll leave it there. And I'm hoping to get more presenters, and we'll have Dr. Tümer come back with an update at some point as well. In the near future, every month or every couple months, we'll be doing another webinar series. Thank you.
Thank you so much. Bye-bye.
So Mara, I think you have to unmute yourself.
There we go. I got the prompt
Okay,
now.
we can
Sorry
hear
about
you.
the delay. Yeah. So just I want to thank Dr. Tümer and Amanda again for all their work. I want to thank you all for coming. The more you understand, the better you understand what's happening, the better for both you and the foundation and the disorder as a whole. And yes, we have opportunities for participation. So when I post about census data and we ask you to give information about that, this helps researchers know where patients are and helps us connect them with appropriate patients if they need to. When I post about Dr. Tümer's research, now you know how she uses it and what she does, and how all those opportunities are very collaborative between the population. They tend to stem from what we ask researchers to look at. Therefore, it's about a better way of life, a better quality of life for our children. It's the symptoms that matter to us. Most of these efforts are collaborative and sort of triggered by us. We, constantly connect with researchers and ask them to study. We really need to support those who agree to. Rare diseases are hard to study because of the numbers, and there are thousands of them. So researchers, we have to tell them what to focus on and who to focus on. And so if we want to be the ones that they're focusing on, we need to let them know we're here and we need to collaborate with them. And one of the big ways, unfortunately, that we also have to work on is fundraising, because governments can't all fund research on these thousands of disorders. And so a lot of it comes from fundraising that the foundation does. So anytime we appeal for your support, we hope you can find small or big ways to contribute, whatever they are. And if you'd like to get involved with the foundation in any way, shape, or form, contact us at any time. We're a fun group. We work very collaboratively. We're pretty friendly. We're very open. And the more of us, doing all of this, the further we'll go.
Thank you so much to everyone for coming, and we'll see you next time. Enjoy the rest of your weekend. Bye.
Thanks, everybody.